Good morning. Myself, Dr. Shobha from Maharani Science College for Women, Mysore. So today's topic of discussion is important discoveries in biochemistry. Now, before I go to the actual topic, let me start with an interesting quote by Frederick Sanger from his Nobel banquet speech. It says, "Scientific research is one of the most exciting and rewarding of occupations." This find interesting to me because we know that science influences most aspects of day-to-day life. It improves human life at every level from individual comfort to the global issues. So, let us now know some of those important discoveries in biochemistry and appreciate them, and also let us thank the scientists for their contribution to understand the subject. Now coming to the subject of biochemistry, we know that biochemistry is the science which deals with the study of chemical basis of life or the chemistry of life. It actually explains the structure and the functions of various molecules that occur in the living cells and organisms, and also explains the chemical reactions that takes place in the living cells and organisms. So all those physiological process, what you know, like digestion, absorption, respiration, excretion, and many more process of the living organisms are all explained in the language of chemistry. In fact, the term biochemistry is used first time by Carl Alexander Newberg in 1903. He is an early pioneer in biochemistry and is also known as father of modern biochemistry. Actually, the beginning of biochemistry cannot be very well said because it is an interdisciplinary subject comprising of organic chemistry and biological sciences. Hence, the beginning of research in the field of life processes can be considered as an advent of biochemical age. It was in 1777, Antoine Lavoisier, a French chemist, he explained the role of oxygen in combustion. He concluded from his experiment that oxygen is consumed in the reaction, carbon dioxide is eliminated, and heat is evolved. This discovery served as a basis for the respiration that occurs in the living organisms also. And from this discovery, for the first time, a physiological process was explained with reference to a non-living mechanism. Another important discovery is discovering the vaccine for smallpox by Edward Jenner in 1796. I can say this was a life-saving discovery because smallpox was a deadly illness which killed around 300 million to 500 million people around the world in the 18th century. But after this vaccine is discovered, the disease is completely destroyed and no traces of smallpox is found now. Next interesting discovery was synthesis of urea by Frederick Sanger in 1828. This urea, which is an organic compound, was synthesized from inorganic salt, that is from ammonium cyanate. So thus, this in vitro synthesis of urea disproved the theory of vitalism, which says that organic substances can only be produced from living things. Hence, many science historians believe that this in vitro synthesis of urea by Euler as the starting point of biochemistry. Now you know that urea is a nitrogenous waste product which is formed in the liver as an end product of deamination of the amino acids, and it also serves as an important role in metabolism of nitrogen containing compounds by animals and is excreted in the urine of mammals. Next, let us know about the contribution of Louis Pasteur, a French scientist who is both the chemist and microbiologist. His contributions are among the most valuable in the history of science, and he is equally claimed by both chemistry and microbiology. Here I'm going to talk about only the vaccine that was developed by Louis Pasteur for anthrax and the rabies. Anthrax is a major livestock disease which affects the animals, and humans can also become affected through contact with the infected animal or by inhaling the bacterial spores. Similarly, the rabies, according to the WHO, is listed as one of the world's most deadly infectious disease, and rabies kills an estimated 160 people a day, or one person for every 9 minutes are killed by the rabies. Therefore, discovering vaccine for these diseases, that is anthrax and rabies, saved the lives of people, and that is why it is considered as a greatest achievement. Then it was Edward Buckner who demonstrated fermentation in the cell-free extract in 1897. This was the first demonstration of a complex biochemical process outside the cell. And he is also a winner of Nobel Prize in Chemistry in 1907. In fact, his work involved the carrying out the fermentation. He observed that yeast extract without living cells can form alcohol from sugar solution. The conclusion of this experiment was biochemical processes do not necessarily require living cells, but they are catalyzed by special molecules called enzymes, and he named this as a zymase. Then this discovery showed almost every significant life process is dependent on the enzyme activity. So enzyme as a catalyst was discovered by Edward Buckner in 1897. One of the greatest discovery of 20th century is discovery of insulin. This insulin was discovered by Banting and Best in 1921. We know that insulin is a peptide hormone which has a role in maintaining blood glucose level, and the deficiency of insulin leads to diabetes. Though this disease is known since 1500 BC or from ancient days, an adequate treatment was not discovered until 1920s. The only treatment available was quit all carbohydrates and live only on the fat and proteins and await death. Hence, this discovery ranks the greatest discovery because it saved the lives of millions of diabetes patients. So, Banting and Best are the world's most celebrated medical heroes. It was in 1929 Carl Lohmann discovered ATP, a molecule called as energy currency of the cell, from the extract of liver and muscle cell. This is called energy currency of the cell because all living cells utilizes ATP for their reactions, and this discovery gave us an understanding the form of energy the cells utilizes. We know that biochemistry does not just explain only the structure and function of various molecules, but it also explains various reactions or the pathways that takes place in the living organism. So one such metabolic pathway that was discovered for the first time was urea cycle. It was Krebs and Hanslet together in 1932 discovered urea cycle. This discovery gave an indication that some of the metabolic pathways can be arranged in cyclic manner also. So they need not be just a linear pathway, but it can also be a cyclic pathway because this urea synthesis of urea is cyclic. This discovery is significant because it serves as a major source of amino acid arginine, and also this ammonia, which is highly toxic molecule, is converted to a less toxic urea for its excretion in the urine. Then it was in 1937, that is exactly 5 years later after the discovery of urea cycle, Another important metabolic pathway, that is citric acid cycle or TCA cycle, was discovered by Hans Krebs. Hence it is also called as Krebs cycle. This discovery is significant because the citric acid cycle pathway is central to nearly all metabolic reactions and is also the source for almost two-thirds of the energy derived from the food in higher organisms. So, the citric acid cycle discovery gave an understanding of the cellular functions or a basic understanding of the cellular metabolism. So after the discovery of two metabolic pathways, that is urea cycle and citric acid cycle, the next metabolic pathway to be elucidated was glycolytic pathway. It was by the pioneering contributions given by Emden, Meyerhof and Pernos in 1940. So that is why the glycolysis is also known as EMP pathway. This discovery actually gave an understanding of how the glucose is oxidized or metabolized inside the cell, and this is an oxygen independent metabolic pathway, and also it might be probably the earliest metabolic pathway to be evolved because this glycolytic pathway is used by nearly all the organisms present on earth for their source of energy. A major breakthrough in the subject of biochemistry in the 20th century came when Watson and Crick proposed the three-dimensional structure for the DNA, which is the genetic material and also known as the molecule of life. And this structure was actually proposed by the evidences given by the earlier research work of Frederick Sanger, the Chargaff, and other scientists. In fact, this structure, the primary structure of DNA, which is a double helical model, helps in understanding the function of DNA as a carrier of heredity or a carrier of genetic material. Hence, this is again a greatest achievement or one of the important discovery in the biochemistry. So, after knowing the structure of nucleic acid, next it was in the field of protein chemistry where Frederick Sanger, a British biochemist, sequenced the primary structure of insulin, a hormone. So, primary structure refers to the order of amino acids in which it is arranged in a protein molecule. This method was automated. This method is also called as Sanger's method and is automated and is used to sequence the human genome in the Human Genome Project. The primary structure of any protein is important because that is the one which determines the tertiary structure or the final folding of a protein. And this tertiary structure again is important because this actually determines the mechanism of action of the protein, its relationship to other proteins, and also its function. Hence, the discovery of the structure of insulin by Frederick Sanger also is a very important in the field of biochemistry or in the field of protein chemistry. Then in 1964, an Indian-born American biochemist, that is Har Govind Khorana, succeeded in synthesizing a polyribonucleotide with a defined repeating sequence. And this polymer is used as a template in the protein synthesis also. Then again, along with the Nirenberg, he elucidated the complete genetic code. So each of the amino acid have a specific codon for them. And the function of these individual codons coding for the respective amino acids in protein synthesis was elucidated by Har Govind Khorana and also Nirenberg. As our understanding of the structure and function of various biomolecules were made, the researchers started an attempt to sequence all the 3 billion base pairs of the human genome. And this work is called as Human Genome Project. This is an international research effort to determine the DNA sequence of the entire human genome, which started in 1990 and completed in 2003 under the leadership of the Francis Collins, support from the US Department of Energy and also NIH. And coming to the importance or the significance of this HGP or the Human Genome Project, by studying the similarities and differences between human genes and those of other organisms, it is possible to discover the function of a particular gene and identify which genes are critical for life. And this knowledge of HGP also has a greater impact in the field of medicine, where better management of the diseases can be done, and also it helps in the early detection of certain diseases and to produce an effective drug or a discovery of effective drugs, and thus totally it helps for a healthy living. Many noteworthy scientific discoveries took place in the 21st century also, and one among them is the chromosome protection by telomere and telomerase, for which a Nobel Prize was given in 2009 jointly to three of the scientists for the research on DNA telomeres and the impact of telomerase enzyme on our cells. So what is this telomere then? So telomere is a repetitive nucleotide sequence at each end of the chromosome. It protects the end of the chromosome from the damage or from fusion with the neighboring chromosomes. But this telomere length decreases or shortens every time the cell copies itself or during the replication. Hence, the telomere serves as an aging clock for each cell. Now here comes the function of telomerase, an enzyme containing two components, a protein and RNA. The function of this RNA component of the enzyme is it serves as a template and synthesizes the telomere DNA, thereby maintain the size or the length of the telomere. So this discovery threw light on the aging process and also on the onset of certain diseases. Like this, the number or the list of discoveries continues to provide us a better understanding of the subject and also a better living for the mankind. And we know with each new discovery, we gain new insight. So the future discoveries are like, of course, there is an ocean of undiscovered findings yet to be solved, but among them, the discoveries which included are effective treatment for various diseases and genetic disorders and formulating rational design of new drugs. So hoping for an effective vaccine for the novel coronavirus which is pandemic at present, I need to conclude this topic with the quote by Stephen Hawking. Scientists have become the bearers of the torch of discovery in our quest for knowledge. Thank you.